Hey everyone, if you're a PharmD or life science student getting ready for exams, then you are in exactly the right place. We're going to cut right to the chase and give you a high-impact review of the absolute core concepts in biotechnology and recombinant DNA. Let's get right into it.
So, here's our game plan. We'll start with the big picture. What even is biotechnology? Then, we'll zoom right into recombinant DNA, talk about the tools you need to make it happen, and walk through the process step by step. After that, we'll see how it's used in the real world and wrap up with a super quick summary of the key facts you absolutely have to know.
All right, first things first. What exactly are we talking about when we use the word biotechnology? It's a pretty broad term.
Well, the official definition from the European Federation of Biotechnology says it's all about integrating natural science with organisms, cells, and molecules to create useful products and services. You know, basically, it's using living things or parts of living things to make stuff that we need.
Okay, so from that huge field, let's zoom in on one of the most powerful techniques out there, recombinant DNA technology. This is really the heart and soul of modern genetic engineering.
So what is it? Recombinant DNA, or rDNA for short, is basically a DNA molecule that's been artificially stitched together. You're taking genetic material from different sources, say a human gene and a piece of bacterial DNA, and combining them. Then you pop this new hybrid DNA into a host, and voila, you've created brand new genetic combinations that didn't exist before.
Now, to do this kind of incredible genetic surgery, you can't just use any old lab equipment. You need a very specific set of molecular tools. Think of it like having a toolkit for editing the very code of life.
And in that toolkit, there are 4 absolutely essential items. You've got restriction enzymes, those are your scissors. You have vectors, which are like the delivery trucks. Then there's DNA ligase, which is basically molecular glue. And finally, you have your host cells, which act as tiny biological factories. Let's take a quick look at each one.
First up, restriction enzymes. These things are amazing. They are proteins that act like super precise molecular scissors. They scan along a DNA molecule and only make a cut at a very specific sequence, called a recognition site. This precision is what makes the whole process possible.
Next, you need a way to get your gene of interest into the host cell. That's where a vector comes in. The most common vectors are plasmids, these little circular pieces of DNA that bacteria have. Now, for a plasmid to be a good vector, it absolutely has to have something called an origin of replication. That's the start signal that tells the host cell's machinery to make copies of it. So, once you've cut your gene and you have your vector ready, you've got to join them together, right? That's the job of DNA ligase. If restriction enzymes are the scissors, then DNA ligase is the molecular glue. It creates permanent bonds and seals that foreign gene right into the vector DNA. And the final piece of the puzzle is the host cell. This is usually a simple bacterium, like E. coli. We put our newly made recombinant DNA into this host, and the host becomes our little factory, making millions of copies of the new DNA and, more importantly, using that DNA to churn out the protein we want.
Okay, so we have the toolkit. Now let's put it all together. How does the process actually work? Well, it follows a really logical 5-step sequence. The 5 big steps are, first, isolation, where we get the gene we want. Second, cutting both the gene and our vector. Third, ligation, where we glue them together. Fourth, transformation, to get the new DNA into our host. And finally, selection and expression, so we can grow the successful cells and get our final product.
So, step 1 is isolation. It's pretty straightforward. We have to break open the source cells and purify the DNA to get a nice clean sample of the specific gene we're actually interested in.
Step 2 is cutting, and this part is super important. We use the exact same restriction enzyme to cut both our gene and the plasmid vector. Why? Because doing this creates compatible sticky ends on both pieces of DNA, which allows them to fit together perfectly like puzzle pieces. Next up, step 3, ligation. This is where our gene of interest naturally slots into the opened-up plasmid. Then our molecular glue, DNA ligase, comes in and seals everything up. This is the moment we officially create our brand new recombinant DNA molecule.
Okay, step 4 is transformation. We've built our custom plasmid. Now we need to get it inside the host bacteria. We usually give the host cells a little bit of a shock, maybe with heat or electricity, which makes them temporarily porous and allows them to take up that new plasmid DNA.
And finally, step 5, selection and expression.
Look, this process isn't 100% efficient, so we need a way to find the bacteria that actually took up our plasmid. A clever way to do this is to include an antibiotic resistance gene on our vector. Then, we grow all the bacteria on a medium that contains that antibiotic. Only the bacteria that's successfully transformed will survive. We take those survivors, grow them in huge batches, and let them get to work producing our protein.
So why do we go through all this trouble? What's the point? Well, the applications of this technology have completely changed medicine and industry.
In medicine, it's a game changer for diagnostic tools and gene therapy. In pharma, it lets us mass-produce critical proteins like insulin, and it's how we create safer vaccines. The hepatitis B vaccine, for instance, was the very first recombinant vaccine ever approved for humans. And in industry, we use it to engineer enzymes for all sorts of things, from making cheese to producing biofuels.
All right, let's bring it all home with a super fast, high-yield summary. If you're studying for an exam, this next part is what you'll want to tattoo on your brain.
Okay, here are the absolute must-knows. First, the process order. Isolation, cutting, ligation, transformation, selection. It's a logical flow. Second, your key players. Restriction enzyme equals molecular scissors. DNA ligase equals molecular glue. And your vector, usually a plasmid, is the delivery truck. And the key fact about that vector? It must have an origin of replication. That's the signal to the host cell to start copying, ensuring our gene gets passed down.
And that covers the essentials of recombinant DNA technology. Best of luck with your studies.
Comment your next topic and subscribe MedX Lab.